Hello, everybody. Welcome to our webinar with Computomics and LAN today. A warm welcome from Tübingen, Germany. My name is Martin Winter. I'm the founder of the Lab Automation Network, and today we will have our webinar together with Computomics, also based in Tübingen, which is new for our webinar series. And today's topic is visual exploration of pangenomes for crop and pharmaceutical science with Pantograph. And Pantograph is a special software from Computomics that Jörg will introduce to you and show you in a live demo today. As a quick introduction from my side, the webinar series Technology Selection 2021 collects outstanding technologies in the field of laboratory automation and digitization. And this is our special area, our business area as the Lab Automation Network. We have different topics: robotics, systems integration, software development, analytical technologies, digital technologies, and so on. And as I said before, today we will focus on Computomics Pantograph software. So for the agenda today, we have, after my introduction, a quick introduction of Computomics, led by Jörg Hagmann. And then we will have the presentation and the live demo of the Pantograph. And after that, there's enough time for questions from your side. Please feel free to ask your questions. Use the Q&A function of the Zoom webinar. Just type in your questions and we will then come up with answers at the end of the session. All right. So to introduce our guest for today, it's Dr. Jörg Hagmann from Computomics. Jörg studied bioinformatics at the University of Tübingen, same university that I come from, but in his case, bioinformatics, and he did his PhD on next generation sequencing, epigenetics, and pangenomics. And I've introduced myself already. I studied chemistry, organic chemistry at the University of Tübingen a long time ago, because for 20 years, more than 20 years now, I'm working in the field of lab automation. So a quick word about the Lab Automation Network. We are an alliance of technology companies in the field, offering, as I said before, outstanding technological solutions like robotics, analytical technologies, software, and all this stuff. And Computomics is a guest today. We are working together closer and closer now, and maybe in the future you will see Computomics, the Computomics logo here on this screen as well. This is just a quick overview about our applications, our technologies that we use, our services. Please feel free to contact us at any time if you have questions about one of those topics or have a project where you need assistance. You're always welcome, and for our customers it's free of charge, because we are paid by our network member companies, our technology providers. All right, and with this I would like to hand over to Jörg and his presentation. Jörg, welcome to the webinar, and the stage is yours.
Thank you very much, Martin, and thank you very much for having us, Computomics and me, and giving us the chance to present our latest developments in the field of pangenomics. Can you see the screen? Yes, good. All right. Yeah, let's start with a few words, as mentioned, about our company, Computomics. So we are a bioinformatics company based in Tübingen, Germany as well. And our main goal is to bring machine learning and latest research technology into industry. We were founded pretty exactly 9 years ago by leading scientists in the fields of machine learning, plant research, and bioinformatics, and completed over 180 projects throughout the world up to now. And our main focus maybe is, or the dominant focus is to improve plant breeding and to accelerate plant breeding. A large product is our Exceed Score technology, which applies genomics prediction by using AI technology. Apart from that, we also have a very comprehensive metagenomics pipeline with user interface and interactive plots to study, for example, plant microbial interactions. And we focus also on pangenomics, the study of multiple sequenced genomes to each other to explore the global genetic variation within a species or population. So overall, these fields pretty well align with our skills: machine learning, plant research, bioinformatics, and help mainly plant breeders to improve their programs. With that, I'd like to switch to the pangenome research. And first of all, I'd like to introduce you to the field of pangenomes by outlining the major benefits of using a pangenome over the more classical approaches when you compare newly sequenced lines against only a single reference genome. I quickly explain pangenome graphs and then move straight on to the new visualization concept we developed. And then I'd like to show a quick demo on a soy pangenome and a quick insight into cannabis pangenome. All right, so as I mentioned, traditionally, if you have sequenced a new organism with next generation sequencing, you compare it against a single reference genome. But now in the last recent years, more and more number of pangenome studies were published. So almost for all the major crop species, there have been sequencing efforts to sequence the full-length genomes of multiple individuals, meaning the pangenome. And also in the human field, there was founded the Human Pangenome Reference Consortium, which really targets towards the replacement of the single human reference genome by a pangenome reference. So these efforts are spurred by really major advances in sequencing technology. The long-read sequencing technology is now more and more affordable to many labs and industries, and those have the great benefit that the read lengths of the sequencing parts are now up more than 200-fold larger than traditionally these short-read sequencing reads. They had still a lower accuracy, but now in the recent years, PacBio hi-fi read sequencing came up, and this remedies the disadvantages of the accuracy. And so with those technologies being more and more widely applied, longer and much more accurate whole genome assemblies are possible. So on the right side, you see for many plant species, the bar plots indicate the number of whole genome assemblies. And on the right side, you see with the blue dots, basically it's a measurement of the assembly completeness. Blue dots indicate these assemblies were made with long-read sequencing data, and green dots with short-read sequencing data. And you see that for basically all the plant genomes, these long-read technology boosted the completeness of the whole genome assemblies. And so what do we do with these whole genome assemblies, and what are the benefits of pangenome studies? In these recent studies, you see that, for example, in 3,000 rice lines, they could identify more than 10,000 novel full-length genes that were not represented in the single reference genome. In pangenome study of soy lines, they found that 2/3 of all the genes present in the reference genome are missing in at least one of only those 26 analyzed soy lines. So meaning a huge diversity in here as well. Pangenome studies also enable the identification of a vast number of structural variants. Those are longer variants. So typically we look only at single base pair nucleotides. But now with complete genomes, we can access also very large pieces of the DNA missing or inserted in, compared to other genomes. And in 100 tomato lines, they found over 200,000 novel structural variants. And in the human field, from 900 human samples, they found an additional sequence to the reference genome of 300 megabases. And this is 10% of an individual human genome. So these variants, that can be now accessed more and more, they are also often associated with phenotypic traits, or more and more studies come up showing that. So here, just a showcase study in Brassica napus, they analyzed the QTL, and in here in the dot plot, you see on the left side, they analyzed one QTL region where the most associated variant was actually an 800 base pair insertion into a gene. And on the right panel, you see that this insertion caused significant gene expression changes. They analyzed a very important trait in plants breeding, namely flowering time. So by analyzing pangenomes, multiple reference genomes, you get a much richer resource for market development, not only focusing on small scale variation, but also on the very big ones and complex ones. This, with more and more variation, you can screen, and you of course also increase the likelihood that these variants are associated to traits or to diseases in human studies. With this, you can find, for example, also completely new traits. For example, resistant genes against stress are found to be in genes that are not present in the reference genome. Additional benefits are that you don't have a reference bias when analyzing your sequenced material, because if you compare it to a single reference, then you will find only variation that is very similar to the reference genome. What is coming up since very new, is that you can now have an increased accuracy of variant calling and genotyping with pangenome graph. So the idea is that you build a graph with high quality data that represents short and long variation and complex variation, and then you can genotype those variants with lower quality data like short reads, because you know now the variants, and you can look for that in these low quality material. That was not possible before with only short reads. And finally, you have also, for a pangenome, you have, if you store multiple individuals, you have all the information, or the information about this population species, in a single data structure. And this data structure, of course, can be compressed, and that is typically nowadays a pangenome graph. So that is basically You have, if you compare several sequences to each other, the classical way is to look at multiple sequence alignment you see on top, where you put the same sequences underneath and different sequences next to each other, basically. And this can be translated into a genome graph, which then stores also the common sequences only once in nodes and alternative sequences separately. And then you can connect the sequences based on the input genomes with these paths shown here. You directly see that this is a compression because we now store much less sequence, right, compared to the multiple sequence alignment. And this is nowadays also spurred by major advancements in algorithmic or in the bioinformatics field. So this development was spearheaded by the Human Pangenome Reference Consortium. They develop tools to generate pangenome graphs, and that also very efficiently and very comprehensively. So nowadays, with, for example, the PGGB software, Pangenome Graph Builder software, you can generate pangenomes of even large and complex genomes within days now, which was not possible actually 2 years ago. And PGGB also, so there are 3 commonly used tools I presented here, and they have differing requirements as input material. PGGB, I would say, is most flexible in taking in being able to build graphs from high quality data like whole genome assemblies, long read data, but also with short read data. PGGB has also the benefit that it's completely reference free, and it basically performs multiple sequence alignments on a regional scale, which basically linearizes your resulting pangenome graph. I should mention why we don't do just multiple sequence alignment on the whole chromosome genome level. That's basically just easily because you cannot do that efficiently. The computational resources don't allow that. And what do you do with the graph? Typically, we have 2 major graph applications. The one is genotyping, which I mentioned quickly in the introduction. And the second one is to visualize the pangenome graph, to explore the whole, the variation included in the pangenome graph visually, basically. And that's where our software Pantograph comes in, which I will present in the following. So the visualization concept, so from a pangenome graph to interactively browsing it, it all started with a grant 2 years ago with our collaborator, a sequencing service facility, Cubic, at the University of Tübingen, and together with an international scientific collaboration. And we came up with a concept to visualize pangenome graphs that basically fulfills 3 main goals. We wanted to illustrate all kinds of genetic variation, right? Many tools out there comparing genomes specialize only on, for example, structural variation, but cannot zoom in into on the single base pair level. We also wanted to be scalable to crop pangenomes. Typically, the important crops are also very large and complex. And with these 2 criteria already, we don't see any other software out there to our knowledge. And finally, we wanted to help researchers, breeders, to explore marker trait associations. And that's why we wanted to to show not only the genetic variation, but also all kinds of metadata like phenotypes, disease, drug response, or metabolites or something like that. I will show that later on. So from a pangenome graph, we, which is quite complicated with all these arrows, we thought we represent this graph as a binary matrix. So we introduce a pangenome sequence you see on top of those cells. And this is basically the concatenation of all the sequence present in the genome graph. That's then, of course, an artificial sequence. And the matrix cells, they are filled if the nucleotide in the first genome is present at that location. And in this way, you can easily compare the genomes, and if cells are blank, you know that this sequence is missing at that position. Now a SNP for here in the middle is now represented as 2 consecutive columns, which is at first a bit counterintuitive. But the representation of a binary matrix has 2 main benefits. The first one is that you can use the color space to annotate or denote additional features, like, for example, different kinds of variation. So inversions can be denoted by a different color. Duplications, for example, here, that this sequence is traversed multiple times. Of course, we try to output it linearly, but this cannot, so you cannot show the whole pangenome graph in a linear way. So if there are synteny changes, so swaps of different sequence stretches, then we have to introduce these kind of arrows, meaning at that position we have to follow the link and, for example, skip the sequence in between the link and follow the path from that new position on. Second benefit of the binary matrix is that we can define zoom levels. So if we move through with a window over the matrix, here, for example, we take 5 consecutive base pairs and summarize them. So meaning the first 5 base pairs are all filled. So we take a filled cell in our zoomed out view. And in this way, we get abstraction levels, right? In the zoomed out view, we now miss the information that there is a SNP, but we still retain the information that there are larger variants like this duplication or deletion actually here. And if you move up further and look at the whole chromosome view, here you see 29 soy whole genome assemblies. That is a graph of length 80 megabases. And here you see this highlighted column is now a window of 100,000 bases, basically, so that the whole chromosome fits on the screen. What you see is that in the middle part, you have a lot of white space. This is unaligned or at least missing sequence in most of the genomes. So meaning here you have most variation going on. You see it also with a lot of links going here. So in this higher view, the links are of simplified shape. And that makes also sense, right? This is the centromeric part of soy genome. This is located here and contains the most variation. In blue, you see genic regions. So all these assemblies were also annotated. And that also makes biologically sense. All the genes are mainly found on chromosome arms of soy genomes and of many other crop species. So if we zoom in a bit on each line, we see that we have for each genome, we have 2 stripes. One is the genome annotation below, and on top It's the coverage, basically. So how many bases are covered in that bin. And with that, you already can see on this large zoom level that there is a larger variation going on. And I mentioned Pantograph was designed so that we can associate traits with genetic diversity. So Pantograph allows the inclusion of a lot of metadata, user-defined metadata, which is shown at a heat map in front of the genome names. And so let's jump into a live demo. I picked a data set of these 29 soy assembly genomes, and we will look into a region that is associated with a trait, namely the seed coat color. So you see that the authors of the manuscript did a hierarchical clustering and saw that 5 lines have a black seed coat color and the rest have a yellow or too transparent seed coat color. They basically categorized all these lines into 5 different haplotypes based on their genetic diversity. So if I switch now to the Pantograph, this is the user interface, and if we load now the soy data set, we can jump now. We can navigate in this pangenome. We can jump, for example, directly to a gene of interest in this region. And I have to say this region is, of course, associated with the trait. So here, if I zoom out a bit, we see now that we have many bases deleted in a few samples, right? If we want to see which samples exactly, we can now also sort by genetic variation here in this region, and then we have all of them underneath. And here we see the first 5 samples, genomes, they have this large deletion. What you also see in, as I told you, the dark blue stripe is a gene, and here it's an exonic, the exonic part. In light green, light blue is the intron sequence. So this deletion, actually, if I click on it, we see which gene it is. This deletion actually disrupts apparently this gene because the whole exon 1 of that gene is missing. Only exon 2 might be present because it's aligned here. So it can have a major impact on traits, for example. And now we can correlate the deletion, this variant, with the metadata. You see in the second column, the metadata for the seed coat color. So you see all these 5 first samples have black seed coat color, all the others yellow ones. Now, and so yeah, this hopefully helps in evaluating. If you look at QTLs, you can evaluate if a variant is really nicely correlated with phenotype of interest. Now we can zoom in again on a single nucleotide level. Here, for example, we have another deletion in these 5 lines. And we see, if I now switch to this bit dense view, if I switch to this matrix kind view, you see that there are a lot of SNPs nearby this large deletion that are also very well linked to this deletion, right? So, for example, if you have the use case that you find this structural variant and you want to design a marker for that for your genotyping panel, for example. Typically, genotyping panels are done with only single SNPs, and so you could select now one of those perfectly linked SNPs to this deletion for your panel, for example. So this also helps in marker selection. And if I move out, so here I wanted to highlight another deletion. If you are interested, how does the genetic variation correlates with a trait, we can also sort the rows by those haplotypes I showed here, the haplotypes that the authors classified here, and to make this more clear, also color the lines by these haplotypes. And then you see that the larger deletion is private to only 3 haplotypes out of the 5. Haplotype 2, 3, and 4. And the other haplotypes, they have only a smaller deletion, actually. So this also should hopefully quickly explore the associations between genetic material and metadata of any kind. What you also can see, it also serves well to compare different gene annotations. So here you see a lot of differences between genes, and they were all de novo annotated, these genomes. And if we are now interested, for example, here, why this gene ends here, we can zoom in to single base pair level again. I switch on the matrix view again, and then you see that this gene ends here, and the last 3 bases are TGA, right? And that is a known stop codon. So apparently, this genome might have a premature stop codon at that position. And if you look here, you spot a missing base just a bit upstream. That could lead to a frame shift and to this premature stop codon. So that is basically the concept of Pantograph, that you can explore single base pair variation, but also larger structural variation if you zoom out, and that you have the entirety of genetic variation that is present in your population at your hand. With that, I switch back to the presentation, and I show you one additional use case in cannabis breeding. Cannabis now establishes as a very important crop for fiber production, but also for medical purposes, right? Very important trait is the ratio between the chemicals THC and CBD. THC is the psychoactive compound of cannabis, but CBD has great potential for medical purposes. And they are regulated by approximately the same enzymes, the same molecular pathway. But the problem for an easy target for breeders is that those genes occur multiple times, very, very often in the genome. And so it's desirable to, of course, understand the mechanism and link the duplicated genes basically to the THC CBD concentration in different plants. And so with Pantograph, I showcase only just one of those many synthase genes, and you can color code the number of traversals of those genes. And here you see, for example, for the reference genome, the 3rd line, a gene, and this is traversed 5 times. So the duplication level of that gene is 5, meaning that sequence occurs 5 times in the genome, but was only annotated 1 time. And by comparing it with other lines, so we made a pangenome of all the 13 publicly available cannabis assemblies. In the other lines, you see quite different duplication levels, right? From 3 up to 10. And for this sample here with the 10 duplications, you also can get from the tooltip that 4 are inverted. So they are switched around, basically. So here, that can also aid in linking duplication levels, maybe to metabolite concentrations or other phenotypic traits. So with that, how much time? Yeah. With that, I want to close and summarize. So I hope that with the demo, I covered some use cases of Pantograph, namely finding or evaluating basically the associations between genetic variants and phenotypic traits by aiding marker discovery. So we can now look at SVs, structural variants. We can look at nested variation, that is variation present in sequence that is not available in the reference genomes. Pantograph lets you help you in fine mapping if you want to pinpoint a causative or correlated variant. Then you can check if that is in a gene region is likely impacting gene function. So you can also compare gene annotations. And what's also relevant right now at the start of the pangenomic research era, maybe, that you can evaluate also the genome graph topology. So you can generate a few pangenome graphs, and Pantograph can read and visualize any variant graph in GFA, in the commonly used GFA or VG format, and you can compare the topology, look if known variants are well represented in these graphs or not. So it's scalable to large pangenomes, so it's independent of the genome size because we're looking only at subsets of the genomes, and the Pantograph is web browser based, meaning you don't have to install any packages, and you can share a view with just exchanging links. So you don't need to load a program, load the data from your hard disk, and then navigate to a position. You can also just navigate to a position just by a link, which you get maybe from your colleague. These are the benefits of Pantograph, and for the end, I'd like to summarize a bit the benefits of looking into pangenome analysis. As I maybe already mentioned, so you get a better understanding of the genetic diversity. You can profile screen a lot more genetic diversity than by only looking at a single reference, so that you get the entirety of the genetic repertoire of your population, of your breeding program, or of the whole species. It aids in trait dissection, and with that, you can improve marker selection and your breeding program, and with that, accelerate crop improvement. So Computomics is here to provide you services. We are very happy to assist you in generating, for example, pangenomes if you have suitable data present. In the pangenome browser Pantograph, we would ask for commercial use and yearly annual license fee, but we're very happy in basically going together with you in researching what is possible with your pangenomic data by generating pangenome graphs, or even exploring graph genotyping on it. So just drop us a line if you're interested, if you have suitable data. And with that, I'd like to close, and I'm very happy and curious to your questions and comments you might have.
All right, thank you so much, Jörg, for this great talk and the demo. Quite impressive. Of course, as expected, we have a lot of questions. So let's quickly jump into it. First one would be, thanks for the presentation. Yeah, also from one of the participants. He does have multiple questions. The first one would be, is there any demo available somewhere? The only one I found was the genome graph version for COVID data, which is a bit old compared to the one you demoed today.
Yes, absolutely. Yeah, that was basically the outcome of our international scientific collaboration, the SARS-CoV-2 browser. But we figured that this is not scalable enough for larger genomes, not very modern, and not very interactive. So we basically took a similar approach than that one, but implemented it anew. Soon we'd like to publish or make the instance available that I showed. So the 29 soy genomes we can host and make it available for browsing for the public. If you're interested, just drop us a line and then we keep you in the loop when this happens.
Yeah, next question from the same participant. He's also interested in the technology used and performance with big data sets. Do you use WebGL, Canvas? And have you faced issues with high number of genomes, let's say hundreds rather than a dozen, for example?
Yeah, right. It's a very technical question, but yeah, the front end is developed with WebGL. Yes, otherwise we cannot do the smooth experience here. Yeah, and that uses graphic cards, CPUs, rather than the normal CPUs, and that accelerates the whole computation. That's right. Yeah, the number of genomes is a bit of a limiting factor. But because we can control how much data we load into one view, we can control also that. And sure, if 10,000 genomes would likely slow it much down. That's true. But there we have also ideas to have kind of vertical summarizations of the genome graph, because for 200 genomes, you cannot also scroll down, up and down. So we thought on our list is basically that you focus on only the variants and highlight the paths that have these variants, basically. So yeah, it's a good advancement we have to think about when pangenome studies get larger and larger.
Mm-hmm. Okay. Then another participant is asking, I'm curious, what format is used as an input for Pantograph? Is it a multi-sequence alignments like FASTA format, or does it use graph database like Neo4j? I'm not an expert in this, but you may know what he means.
Very skilled audience, yeah. No, as I quickly mentioned, maybe it uses graph format, so this GFA format, or from the VG toolkit, this VG format. And that is basically the outcome of the 3 major pangenome generation tools I showed before.
All right, a second question in a similar direction. Does Pantograph accept pangenome graphs generated by any tool? And how much is the license fee for Pantograph?
Yeah, generate by any tool? No, actually, I would say maybe, to my knowledge, by any tool that is capable of producing really large pangenome graphs. So as I told, the PGGB output, minigraph, progressive cactus, and VG, those output graphs in GFA or convertible to GFA format. And that is the input for Pantograph. Yeah, license fee, I'm the wrong person, no. We there, please contact us, and then we talk about that.
Okay, maybe in general you could comment on how such a project would work. If somebody comes, ask for license fees, ask for a project, how do you conduct such requests from customers?
Yeah, so we in all our projects we first try to understand completely what the customer's needs are at first, what he wants to achieve, and then how good the data is already that he has. So we would discuss about the goals, the data available, and then we would discuss what is feasible with this data, and then discuss what's best strategy to move forward. Maybe to generate another graph based on different parameters if the available graph might be not suitable for the question, or just try to visualize it. Yeah.
All right. Then the next question, how does Pantograph handle pangenomes built from genomes that are only assembled on contig or scaffold level? And asking, since the examples you showed are all just one specific chromosome.
Yeah, for that we are actually about in implementing that, because also the human pangenome consists of only contigs and not full assembled chromosomes or pseudochromosomes. But there you have then basically different paths per chromosome, and they are basically merged to only a few non-overlapping paths. That is our strategy, because complete merging might introduce contradictory sequences, and so we keep basically the contradictory contigs both, and only merge contigs that don't overlap or not contradictory overlap.
All right, thank you very much. Next question, what's the maximum number of genomes that Pantograph can compare?
Yeah, that depends on a bit the data, and there are a few parameters that we can optimize, so I cannot give a general number here. That's basically have to try out for the specific data set and species, and that can be also tweaked by the preparation script on the back end. So yeah, I think I haven't mentioned from this GFA or VG format, there is one conversion step, but that is also included in current graph manipulation software to be loaded into Pantograph. Yeah.
All right. Then one question that is also in the chat. Are you planning to make the source code freely available?
Greetings to Andrea. So right now the strategy is that we host selected pangenomes that are freely browsable, and that for the community can freely look up the genetic material and experience basically the Pantograph browser. For a full open source, cannot.
Not yet.
Yeah, not yet.
Good. Then two more questions right now, and I think we would still have time for a couple of more. To my knowledge, the graph format holds the information about the genomic structure, paths taken, sequences, but do not have gene annotation within. Could you explain how these information are merged?
Yes, exactly. The genome annotation is separate. We basically use available GFF files that are the typical file format for gene annotations. And in this conversion step, merge that with the graph structure, basically between back end and front end, these gene annotations are read in and denoted to the respective locations. Yeah.
Okay. Last question up to now is the following one. Does it support sequence searching or export?
Yeah, that's very nice features. Sequence searching is not on our list yet, or not a high priority. Sequence export, it absolutely makes sense, and that's on our to-do list to come. Yes.
All right, then just one more coming in. Can you impute breeding population progeny based on a parental pangenome? Oh my God, I don't understand this question, but you surely do.
So, of course, Pantograph can only visualize what's in the pangenome graph, and don't have special kind of interface for imputation tools. However, we right now actually also implement the integration of haploblocks. So from many sequences, you can identify haploblocks. These are regions of local similar local sequence, and they represent basically the blocks that get most often inherited at in one block, basically. When we could integrate such haploblock information quite easily into such a tool, and with having the haploblocks, those can be used as for imputation. So if you have the parental pangenome, you build haploblocks, and then you have to identify from your progenies, yeah, the SNPs within a haploblock. There you can then do imputation, but well, you can only check then in the Pantograph visualization how these SNPs nearby within a single haploblock would then also look like. It doesn't output the imputed genotypes.
Okay, that was the last question from my list, but I have another one. So what are the next steps? What are you currently working on with your Pantograph software?
Yes, I just mentioned the haploblocks are a nice way in abstracting genetic variation, and as just told, also a nice way to impute missing data. That is currently worked on, yeah, then we and then we are always thinking of integrating additional metadata, or like regional metadata, like for example epigenetics or even gene expression data. So you could, for example, if you pick a tissue of interest, you could then color code the genes based on the expression levels, for example.
All right, good. So now we're at the end of the questions, also at the end of this this webinar. Thank you so much, Jörg, for the presentation. Thanks for the good preparation of this webinar to two further colleagues, Simone Betz from Computomics and Stefanie Schulze from Lab Automation Network. And to all the participants, thanks for joining. Please, if you have any further questions or any ideas, reach out to us via LinkedIn, via email. I think you will find a way to reach us, and we would be happy to discuss those topics with you, to work with you. And of course, if you have friends or colleagues that could potentially be interested in the same topics, let them know. We will make the recording of this webinar available to you and also to your colleagues. Just drop us a note, a quick message in LinkedIn or an email. All right. So thank you very much, everybody, and have a coffee or a bottle of beer now. If you are in Germany, now we are close to evening, and yeah, hope to talk to you again soon. Bye-bye.
Bye. Thank you.